In this video, I'm going to be talking about why we need to develop new test methods for the coronavirus. And the issue that we currently have is that people who are hospitalized due to the coronavirus will have their cheek and nose cells swabbed, and this is to detect if you have the genetic material for the virus. And the issue is that these tests that we're running, in which we are performing PCR to determine if you have that viral genetic material inside of your cells, is only checking if you currently are infected with the virus. It does not tell us if you were previously infected with the virus, and this causes a lot of problems in order for our epidemiologists to determine accurate numbers for how much of a response society should have to these diseases. And so if we instead make use of antibody testing, in which we check patient blood samples for any antibodies that will have affinity for the coronavirus, this will help us be able to identify whether or not you've had or currently have infections for the coronavirus. And so why is it important that we get this more reliable data? Well, if we're able to determine which healthcare professionals, like what doctors and nurses and lab technicians have this adapted immunity for the coronavirus, it means that they can be much more exposed to the virus, and we will have more healthcare resources available to us. And right now, in the middle of a pandemic, we need as many healthcare resources as we can get to treat as many patients as we can, because the rates at which this virus is spreading is significantly higher than our healthcare resources currently can sustain, so we need as much as we can get right now. In addition to that, when epidemiologists are trying to determine something referred to as the attack rate, which is the percentage of people in a population who contract a disease, if we don't know how many people have been infected, we don't know, or we cannot calculate the attack rate. And this is an important metric determining for world leaders how big of a response we should have to this disease. In addition to that, if we don't know how many people were previously infected by a disease and are currently living, it causes us to overestimate the fatality rate, which is another thing that we are currently doing with our statistics. So we may be overreacting, and we may be able to get more data in here so that we can have more insights in how to effectively tackle the coronavirus and COVID-19. And so instead of just telling us that we need to do antibody testing, I figured I'd also talk about how would we do it. And with antibody screening, it's the first step in this process is going to be determining which proteins on the coronavirus are conserved. And because the coronavirus is a virus that mutates very often, its genetic material changes, its proteins change, we need to determine what proteins on this virus can we use in order to have effective and reliable antibody screening kits. And so once we've done this, the next step here would be to acquire patient blood samples, and then with those patient blood samples we would do things called Eliza tests. And Eliza tests are used in clinics already for screening for HIV, as well as Lyme disease, among other diseases. And so once we've bound proteins onto the surface of well plates, once we've identified what are the conserved regions or conserved proteins for the coronavirus, put those on these well plates, we've flown the patient blood sample into these wells. If the patient has any antibodies that will stick onto the coronavirus proteins that we have agreed on are valid proteins to use for our clinical testing, then we would flow in a secondary antibody. Secondary antibody would stick onto any primary antibodies which came from the patient that are bound onto the coronavirus, and then finally we would flow in a substrate which is colorless until it interacts with an enzyme on the secondary antibody. And so basically, at a high level, if we see any color change from a patient's blood sample, it would indicate to us that a patient has or previously had been infected with the coronavirus, which would help us have far better metrics for mounting a response at a higher level. And so this is going to wrap things up for this video. I hope you guys find this stuff useful. Let me know if you have any questions. Please stay safe out there and wash your hands, and take care.